The 1860s, a time of profound change. The world was on the brink of a technological revolution. We were grappling with new ideas. The whispers of scientific discovery were in the air. Charles Darwin had just published his groundbreaking work on the origin of species. This book challenged centuries-old beliefs about the natural world. It introduced the theory of evolution by natural selection. Imagine the intellectual fervor of the time. Darwin's ideas sent shockwaves through the scientific community. They ignited a firestorm of debate. But Darwin's theory left a crucial question unanswered. How were these traits passed down through generations? What was the mechanism of inheritance? The answer to this question, as we would discover, lay deep within the cells of every living organism. This is the story of that discovery. It is a journey that begins in the 1860s. It is a story of scientific curiosity, perseverance, and brilliance. It is a story that would ultimately revolutionize our understanding of life itself. Before we delve into the discovery of DNA, we must first understand the scientific landscape of the 1860s. At that time, the concept of genes, as we know them today, did not exist. Scientists knew that traits were passed down from parents to offspring. But the how remained a complete mystery. They observed the resemblance between family members, but they lacked the tools and knowledge to explain the underlying mechanisms. Some scientists proposed the idea of blending inheritance. They believed that traits from parents blended together, like mixing paint, resulting in an average of the two. This theory, however, had its flaws. It couldn't explain the reappearance of traits that seemed to disappear for a generation. The true nature of inheritance was far more intricate and elegant. The scientific world was ripe for a breakthrough. It needed someone with a keen eye for observation, someone who could connect the dots and unravel the secrets hidden within the very fabric of life.
Enter Gregor Mendel, an Augustinian friar with a passion for science. In the quiet confines of his monastery in Brno, Mendel embarked on a series of experiments that would forever change our understanding of inheritance. His chosen subjects were simple pea plants, but the implications of his work would reach far beyond the monastery walls.
Mendel meticulously crossbred pea plants with different characteristics. He tracked the inheritance of traits like flower color, seed shape, and plant height. Through careful observation and meticulous record-keeping, Mendel noticed patterns emerging in the way these traits were passed down. His work laid the foundation for the field of genetics. He provided the first concrete evidence for the existence of discrete units of inheritance, which we now call genes. Mendel's discoveries, however, were met with resistance and indifference. The scientific community was not yet ready to embrace his revolutionary ideas. His work languished in obscurity, awaiting its time to shine on the world stage. Let's take a moment to appreciate the sheer audacity of Mendel's work. Here was a monk, armed with nothing but simple tools and an insatiable curiosity, challenging the very foundation of our understanding of life. He meticulously cultivated and crossbred thousands of pea plants, carefully recording his observations in notebooks that would later become the cornerstone of modern genetics.
Imagine Mendel, hunched over his plants, carefully pollinating each flower and meticulously labeling the offspring. He meticulously tracked the inheritance of traits generation after generation, patiently unraveling the secrets hidden within those humble pea plants.
His work was a testament to the power of observation, dedication, and a deep-seated belief that the natural world held secrets waiting to be discovered. But Mendel's genius extended beyond mere observation. He possessed a rare ability to discern patterns and formulate hypotheses. He analyzed his data with a statistical rigor that was ahead of his time. His work was a symphony of science and mathematics, a testament to the power of human ingenuity. Section 5. Beyond the Monastery Walls. While Mendel's work remained largely unnoticed during his lifetime, the seeds of a scientific revolution had been sown. The world was on the cusp of a new era in biology. Scientists were beginning to unravel the complexities of the cell. The discovery of chromosomes, thread-like structures found within the nucleus of cells, added another layer to the puzzle of inheritance. The scientific community was beginning to piece together the intricate mechanisms of life. The stage was set for the next act in this grand scientific drama. The discovery of DNA, the molecule of life itself, was just around the corner, and with it, a new chapter in human understanding was about to begin. Section 1. The Microscopic World Unveiled. As the 19th century drew to a close, scientists were peering deeper into the hidden world of cells. Microscopes, once crude instruments, were becoming increasingly sophisticated. They allowed researchers to observe the intricate machinery of life in unprecedented detail. Imagine the sense of wonder as scientists observed the bustling activity within a single cell. They witnessed the division of cells, the replication of chromosomes, the intricate dance of life unfolding before their very eyes. The cell, once thought to be a simple homogeneous entity, was revealed to be a complex and dynamic microcosm. The discovery of chromosomes in the late 1800s was a pivotal moment in the journey toward understanding inheritance. These thread-like structures, visible within the nucleus of dividing cells, seem to hold the key to the secrets of heredity. Scientists observed that chromosomes came in pairs, one set inherited from each parent, mirroring the patterns of inheritance Mendel had observed in his pea plants. Section 2. The Dance of Chromosomes. Scientists watched in awe as chromosomes engaged in a precisely choreographed dance during cell division. They observed the duplication of chromosomes, ensuring that each daughter cell received a complete set of genetic instructions. Imagine the intricate ballet of chromosomes as they pair up, align themselves along the equator of the cell, and then split apart, each daughter cell receiving a full complement of genetic material. This elegant process, repeated countless times throughout the life of an organism, ensures the faithful transmission of genetic information from one generation to the next. The observation of chromosomes provided a physical basis for the abstract concept of genes. Scientists began to speculate that these discrete units of inheritance resided on chromosomes, like beads on a string. Each gene, they theorized, occupied a specific location on a chromosome, dictating a particular trait. Section 3. The Hunt for the Genetic Material. The early 20th century witnessed a flurry of research aimed at pinpointing the elusive genetic material. Scientists knew that chromosomes were composed of two main components, proteins and nucleic acids. Proteins, with their diverse structures and functions, seemed like the more likely candidate to carry the complex code of life. Nucleic acids, on the other hand, were considered too simple, too monotonous in their structure to carry such a vast amount of information. Imagine the scientific community divided, each camp championing their chosen candidate for the coveted title of genetic material. The debate raged on, fueled by a combination of experimental evidence, intuition, and perhaps a touch of scientific rivalry. The quest to uncover the nature of the genetic material was not merely an academic pursuit. It held the promise of unlocking the secrets of heredity, disease, and ultimately, the very essence of life itself. The stakes were high, and the scientific world watched with bated breath as the drama unfolded. Section 4. Enter the Nuclein. Our story now takes us back to the 1860s, a few years after Mendel's groundbreaking work with pea plants. In a laboratory in Tübingen, Germany, a young Swiss chemist named Friedrich Miescher was engrossed in a rather unglamorous task, analyzing the chemical composition of pus.
I meticulously collected pus-soaked bandages from a nearby clinic. drawn to their abundance of white blood cells, which I hoped would provide insights into the composition of cells. Little did I know that my meticulous analysis of this rather unpleasant substance would lead me to a momentous discovery.
From the nuclei of white blood cells, Misha isolated a substance unlike anything previously known. It was acidic in nature and rich in phosphorus, unlike the proteins he was familiar with. He named this mysterious substance Nuclein, unaware that he had stumbled upon the very molecule that held the secrets of heredity, deoxyribonucleic acid, or DNA. Section 5. The Transforming Principle. Misha's discovery of Nuclein, while significant, did not immediately solve the mystery of the genetic material. The scientific community remained largely focused on proteins as the likely carriers of genetic information. However, a series of groundbreaking experiments in the 1920s and 1930s began to shift the tide in favor of DNA. In 1928, British bacteriologist Frederick Griffith was studying the bacterium Streptococcus pneumoniae, which causes pneumonia. He observed that a harmless strain of the bacteria could be transformed into a deadly strain by exposure to a heat-killed, virulent strain. This transforming principle, Griffith concluded, must be some substance capable of transferring genetic information from the dead bacteria to the living ones. But what was the nature of this transforming principle? The answer remained elusive, but the stage was set for a series of experiments that would forever change our understanding of genetics. The scientific world was abuzz with excitement and anticipation. Griffith's discovery of the transforming principle had opened up a new avenue of investigation. Researchers around the globe were eager to unravel the identity of this mysterious substance that held the power to transform harmless bacteria into deadly pathogens. In 1944, a team of American scientists led by Oswald Avery, Colin MacLeod and Macklin McCarty decided to tackle this challenge head-on. They meticulously repeated Griffith's experiment, but with a crucial twist. They systematically destroyed different components of the heat-killed virulent bacteria proteins, lipids, carbohydrates and nucleic acids, one by one, to determine which component was essential for transformation. To their astonishment, they found that only when they destroyed DNA did the transformation process come to a halt. The transforming principle, they concluded, had to be DNA. The molecule that Misha had discovered decades earlier, dismissed by many as too simple to carry genetic information, was, in fact, the very essence of heredity. The discovery that DNA was the genetic material sent shockwaves through the scientific community. It was a watershed moment, a paradigm shift that would forever alter the course of biological research. And yet, amidst this excitement, a sense of bittersweet irony lingered. For in the midst of celebrating this momentous discovery, the scientific world was reminded of a forgotten hero, Gregor Mendel.
And my groundbreaking work with pea plants, once dismissed and ignored, is now hailed as the foundation upon which the entire field of genetics stands.
Mendel's meticulous observations, his elegant experiments, his revolutionary concept of discrete units of inheritance, all suddenly sprang to life, illuminated by the newfound understanding of DNA as the carrier of genetic information.
His legacy, once lost in the annals of scientific obscurity, is now firmly cemented as one of the giants of modern biology.
The discovery of DNA as the genetic material marked the dawn of a new era in biology, the era of molecular genetics. Scientists were no longer limited to observing the inheritance of traits in living organisms. They now had the tools to study the very molecule that carried the code of life. The race was on to decipher the structure of DNA, to unlock the secrets held within its elegant double helix. The stage was set for the next chapter in this grand scientific adventure, a chapter that would culminate in one of the most iconic discoveries in human history, the unraveling of the DNA double helix by James Watson and Francis Crick. The discovery that DNA was the genetic material set off a scientific frenzy. Imagine the world's brightest minds, fueled by ambition and a thirst for knowledge, all converging on a single tantalizing question. What was the structure of this enigmatic molecule?
The race to solve the structure of DNA was on, a high-stakes pursuit with the potential to unlock the very secrets of life.
At the forefront of this intellectual clash were two young scientists at the Cavendish Laboratory in Cambridge, England. James Watson, an ambitious American with a PhD in genetics, and Francis Crick, a British physicist with a keen interest in biology.
Watson and Crick, an unlikely duo with a shared passion for unraveling the complexities of life, would become synonymous with the discovery of the DNA double helix. Their journey, however, was not without its share of dead ends, false starts, and fierce competition. Across the English Channel at King's College London, another key player in this scientific drama was making significant strides.
Rosalind Franklin, a brilliant chemist, was pioneering the use of X-ray crystallography to study the structure of biological molecules, including DNA.
Imagine Franklin, meticulous and determined, bombarding DNA fibers with X-rays, capturing the scattered radiation on photographic plates. These plates, known as diffraction patterns, held subtle clues to the three-dimensional arrangement of atoms within the DNA molecule.
Franklin's expertise in X-ray crystallography was unmatched. Her images were of exceptional quality, providing unprecedented insights into the architecture of DNA.
However, her critical contributions to the discovery of the DNA double helix would be tragically overshadowed her story, a poignant reminder of the importance of recognizing the achievements of all who contribute to scientific progress. One of Franklin's diffraction images, famously known as Photo 51, would prove to be a pivotal clue in the race to solve the structure of DNA. This image, captured by Franklin's graduate student Raymond Gosling, revealed a striking X-shaped pattern, suggesting a helical structure with a regular repeat distance.
Imagine Watson's astonishment when he first laid eyes on Photo 51, reportedly without Franklin's knowledge or consent. The clarity of the image, the unmistakable signature of a helix, ignited a spark of inspiration, propelling him and Crick forward in their quest to build a model of DNA.
The significance of Photo 51 cannot be overstated. It provided the crucial experimental evidence that Watson and Crick needed to solidify their understanding of DNA's structure. It was a turning point in the race, a moment where intuition and experimental observation converged to illuminate the path toward a groundbreaking discovery.
Armed with the insights gleaned from Photo 51 and our own deep understanding of chemical principles, we embarked on a period of intense model building.
We meticulously crafted and discarded countless iterations of our model, striving to reconcile the available data with the constraints of chemistry and physics.
Imagine the scene in their cramped Cambridge office, littered with cardboard cutouts, wires, and scraps of paper as they pieced together the puzzle of DNA's structure.
Our model, a thing of both elegance and complexity, gradually took shape, revealing the iconic double helix, two intertwined strands of nucleotides connected by hydrogen bonds between complementary base pairs.
Our model explained the regular repeat distance observed in Franklin's X-ray diffraction patterns, the precise base pairing that held the two strands together, and most importantly, how genetic information could be encoded and transmitted from one generation to the next.
In April 1953, Watson and Crick published their groundbreaking findings in the scientific journal Nature, forever changing the course of biology. Their paper, a mere 2 pages long, described the elegant structure of DNA, the molecule that holds the blueprint of life. Imagine the scientific community abuzz with excitement, marveling at the simplicity and elegance of DNA's structure, a structure that hinted at the molecule's profound implications for understanding heredity, evolution, and the very essence of life. The discovery of the DNA double helix ignited a scientific revolution, ushering in the era of molecular biology. It provided a framework for understanding how genetic information is stored, replicated and passed down from one generation to the next, opening up unprecedented avenues for research in medicine, agriculture and biotechnology. With the structure of DNA revealed, the next challenge was to crack the code it contained. Scientists knew that the sequence of nucleotides, adenine, thymine, guanine and cytosine, held the instructions for building and maintaining an organism. But how was this code read and translated into the proteins that carry out the functions of life? Imagine the scientific community embarking on a global effort to decipher the genetic code, a code written in the language of 4 simple letters, yet capable of orchestrating the symphony of life. This monumental task involved a series of elegant experiments, painstakingly piecing together the rules by which the DNA code is translated into proteins. The discovery of the genetic code, a universal language shared by all living organisms, stands as one of the crowning achievements of modern biology. Section 7. The central dogma, from DNA to protein. The flow of genetic information from DNA to RNA to protein, known as the central dogma of molecular biology, emerged as a fundamental principle of life. DNA, the master blueprint, is transcribed into RNA, a messenger molecule, which is then translated into proteins, the workhorses of the cell. Imagine the intricate machinery within a cell, orchestrated by the instructions encoded in DNA. Genes, segments of DNA that code for specific proteins, are transcribed into RNA molecules, which then ferry these instructions to the ribosomes, the protein synthesis factories of the cell. The central dogma provided a unifying framework for understanding the molecular basis of life. It explained how genetic information is expressed, how traits are passed down from one generation to the next, and how mutations in DNA can lead to changes in protein structure and function, sometimes resulting in disease. Section 8. The Tools of Revolution. The discovery of the DNA double helix and the unraveling of the genetic code sparked a technological revolution in biology. Scientists, armed with this newfound knowledge, developed an arsenal of tools to manipulate, analyze and harness the power of DNA. Imagine the excitement as techniques like polymerase chain reaction or PCR and DNA sequencing emerged, enabling scientists to amplify, sequence and analyze DNA with unprecedented speed and precision. These technological advancements revolutionized fields as diverse as medicine, forensics and agriculture. They paved the way for gene therapy, personalized medicine, genetically modified organisms, and the Human Genome Project, a monumental effort to map the entire human genome. Section 1. The stage is set, a scientific quest. By the early 1950s, the race to uncover the structure of DNA had reached a fever pitch. Scientists around the world were hot on the trail, driven by a thirst for knowledge and the understanding that this discovery held the key to unlocking the secrets of life itself. Imagine the atmosphere in those labs, a palpable sense of excitement mixed with intense competition. Researchers huddled over their benches, poring over X-ray diffraction patterns, sketching potential structures and engaging in heated debates. The air crackled with anticipation, a sense that a groundbreaking discovery was within reach. At the Cavendish Laboratory in Cambridge, England, 2 young scientists found themselves at the forefront of this scientific revolution.
James Watson, a brash American with a PhD in genetics.
And Francis Crick, a British physicist with a keen interest in biology.
Their paths crossed in the hallowed halls of Cavendish, a place where brilliant minds converge to unravel the mysteries of the universe. Watson, eager to make his mark, and Crick, with his infectious enthusiasm and knack for problem solving, formed a formidable team. The key to unlocking the structure of DNA came from an unlikely source, a photograph. Rosalind Franklin, a brilliant chemist working at King's College London, had mastered the technique of X-ray crystallography and captured stunning images of DNA fibers.
One particular image, known as Photo 51, would prove to be a turning point. The image revealed a striking X-shaped pattern, a telltale sign of a helical structure. When I saw this image, reportedly without Franklin's knowledge, I was struck by a moment of profound inspiration. The clarity of the X-shaped pattern, the subtle hints of a double helix, ignited a spark in my mind. I rushed back to Cambridge, eager to share this crucial piece of evidence with Crick. Together, we raced back to our model building kits, energized by a renewed sense of purpose.
With Photo 51 as our guide, we embarked on a final push to solve the structure of DNA. We meticulously adjusted our models, incorporating the insights gleaned from Franklin's image. And then, in a moment of scientific serendipity, the pieces of the puzzle fell into place.
As Watson and Crick celebrated their groundbreaking discovery, Rosalind Franklin's contribution to unraveling the structure of DNA remained largely unacknowledged. Her meticulous work with X-ray crystallography, particularly her iconic Photo 51, had been instrumental in guiding their model building.
Tragically, my brilliant career was cut short by my untimely death from ovarian cancer in 1958 at the age of 37. I never received the full recognition I deserved for my pivotal role in one of the most significant scientific discoveries of the 20th century.
It wasn't until years later, with the publication of Watson's personal account, The Double Helix, that Franklin's contribution began to receive wider recognition. Watson's book, while controversial for its portrayal of Franklin, nonetheless shed light on her crucial role in unraveling the secrets of DNA.
Today, I am rightfully celebrated as a scientific pioneer, a brilliant mind whose contributions to science were tragically cut short. My story serves as a powerful reminder of the importance of recognizing the achievements of all who contribute to scientific progress, regardless of gender or background.
In April 1953, the world caught its first glimpse of the elegant double helix structure of DNA.
Watson and I published our groundbreaking findings in the prestigious scientific journal Nature, forever changing the course of biology and genetics.
Imagine the scientific world buzzing with excitement as news of their discovery spread like wildfire. The elegance and simplicity of the double helix, two intertwined strands of nucleotides connected by hydrogen bonds between complementary base pairs, captivated the imaginations of scientists and the public alike.
The discovery of the DNA double helix was hailed as a triumph of human ingenuity, a testament to the power of collaboration and the relentless pursuit of knowledge.
It earned Watson, Crick, and Maurice Wilkins, the head of the lab where Franklin worked, the Nobel Prize in Physiology or Medicine in 1962.
The Nobel Prize solidified their place in scientific history, but it's crucial to remember that their achievement was built on the foundation laid by many others, including Rosalind Franklin, whose contributions were tragically overlooked during her lifetime. With the structure of DNA revealed, the next challenge was to crack the code it contained. Scientists knew that the sequence of nucleotides, adenine, thymine, guanine, and cytosine, held the instructions for building and maintaining an organism. Imagine the scientific community embarking on a global effort to decipher this genetic code, a language written in the four-letter alphabet of DNA. The race was on to understand how this sequence of letters translated into the complex tapestry of life. The task was daunting, but the scientific community rose to the challenge. Through a series of ingenious experiments, researchers began to unravel the secrets of the genetic code. They discovered that groups of 3 nucleotides called codons, each coded for a specific amino acid, the building blocks of proteins. The cracking of the genetic code was a monumental achievement, revealing the elegant simplicity of life's blueprint. It provided a Rosetta Stone for understanding how genetic information is stored, read and translated into the proteins that carry out the functions of life. The discovery of the DNA double helix and the deciphering of the genetic code marked the dawn of a new era in biology, the era of molecular biology. Scientists were no longer limited to studying life at the level of tissues and organs. They now had the tools to explore the molecular basis of life itself. Imagine the excitement and sense of possibility that gripped the scientific community. The doors to a whole new world had swung open, revealing a vast landscape ripe for exploration. Molecular biology promised to revolutionize our understanding of life, from the smallest microorganisms to the complexities of the human brain. The impact of molecular biology has been nothing short of transformative. It has led to breakthroughs in medicine, agriculture, and biotechnology, paving the way for personalized medicine, gene therapy, genetically modified organisms, and a deeper understanding of the evolutionary history of life on Earth. Section 8. The future unfolds. The enduring legacy of DNA. The discovery of the DNA double helix was just the beginning. It sparked a scientific revolution that continues to this day, pushing the boundaries of our understanding of life and opening up exciting new possibilities for the future. Imagine a future where diseases are eradicated before they even manifest, where crops are engineered to thrive in challenging environments, and where we can trace our ancestry back through time with unprecedented accuracy. The future of DNA science is filled with promise and potential. The enduring legacy of DNA is a testament to the power of human curiosity, the relentless pursuit of knowledge, and the transformative impact of scientific discovery. As we continue to explore the vast landscape of the genome, we can only marvel at the elegance and complexity of life's blueprint, forever grateful for the pioneers who illuminated the path to understanding this fundamental molecule. The rise of DNA technology. The discovery of the double helix structure of DNA was just the beginning. Scientists needed new tools to unlock the secrets of DNA. The race was on to develop techniques to read, edit, and manipulate DNA. The development of restriction enzymes was a major breakthrough. Section 2. The Gene Hunters Mapping the Human Genome. As scientists developed new tools for working with DNA, a bold and ambitious goal began to take shape, mapping the entire human genome. The Human Genome Project, launched in 1990, was an international collaboration aimed at determining the complete sequence of the 3 billion base pairs that make up human DNA. Imagine the scale of this undertaking, a scientific endeavor on par with putting a man on the moon. It involved thousands of scientists across the globe working tirelessly to decipher the genetic blueprint of our species. The Human Genome Project was a testament to the power of collaboration, technological innovation, and the unwavering belief in the transformative potential of science. It was a race against time, a race to unlock the secrets held within our own DNA, secrets that promised to revolutionize medicine, shed light on human evolution, and redefine our understanding of life itself. The project was not without its critics. Some argued that it was too expensive, too ambitious, and that the benefits would not outweigh the costs. Others raised ethical concerns about the potential misuse of genetic information. DNA sequencing takes center stage. The Human Genome Project faced the challenge of determining the precise order of nucleotides in DNA. Early methods were laborious, but new technologies emerged, pushing boundaries. Automated machines and algorithms accelerated the Human Genome Project. The first draft of the Human Genome was announced in 2000. The complete sequence was published in 2003, revolutionizing our understanding of biology and disease. Section 4. Chain reactions and amplifications, the power of PCR. While DNA sequencing provided the means to read the genetic code, another revolutionary technology emerged, polymerase chain reaction, or PCR. Developed by Carey Mullis in 1983, PCR allowed scientists to amplify specific DNA sequences from scarce material. Imagine taking a single DNA molecule and generating billions of copies within hours. PCR revolutionized genetics, forensics, medicine, and archaeology. It earned Carey Mullis the Nobel Prize in Chemistry in 1993. Section 5, the Genetic Revolution, Applications Across Disciplines. The development of powerful new DNA technologies sparked a genetic revolution, transforming fields as diverse as medicine, agriculture, forensics, and conservation biology. The ability to analyze, manipulate, and understand DNA opened up unprecedented possibilities for addressing some of the world's most pressing challenges. In medicine, the rise of genomics led to personalized medicine, tailoring treatments to an individual's genetic makeup. Gene therapy, though still in its early stages, held the promise of correcting genetic defects at their source, offering hope for patients with previously untreatable diseases. In agriculture, genetically modified organisms, or GMOs, emerged as a powerful tool for improving crop yields, enhancing nutritional content, and reducing the need for pesticides and herbicides. While GMOs remain a topic of debate, their potential to address global food security challenges cannot be ignored. In forensics, DNA fingerprinting revolutionized criminal investigations, providing a powerful tool for identifying suspects, exonerating the wrongly convicted, and resolving paternity disputes. Section 6. A new era of medicine, personalized genomics, and beyond. The ability to read the human genome ushered in a new era of medicine, one where treatments could be tailored to an individual's genetic makeup. Personalized genomics promised to revolutionize healthcare, offering the potential for more accurate diagnoses, targeted therapies, and even disease prevention. Imagine a future where your doctor could analyze your genome to assess your risk for various diseases, predict your response to different medications, and develop a personalized health plan tailored to your unique genetic profile. While personalized genomics is still in its early stages, it's already having a significant impact on healthcare. For example, cancer treatments are increasingly being guided by genetic testing, allowing oncologists to select the most effective therapies based on the specific mutations present in a patient's tumor. The rise of pharmacogenomics, the study of how genes influence drug response, is also transforming the way medications are developed and prescribed. By understanding how genetic variations affect drug metabolism and efficacy, doctors can minimize adverse reactions and optimize treatment outcomes. Section 7. DNA, fingerprinting, revolutionizing forensics and justice. The ability to identify individuals based on their unique DNA profile, a technique known as DNA fingerprinting, revolutionized forensic science. Developed in the 1980s by Sir Alec Jeffreys, DNA fingerprinting quickly became an indispensable tool for law enforcement agencies around the world. Imagine the impact of this technology on criminal justice. For the first time, investigators had a powerful tool to link suspects to crime scenes, even in cases with little or no other physical evidence. DNA evidence proved particularly valuable in sexual assault cases, where it often provided the key to identifying perpetrators and bringing them to justice. But DNA fingerprinting's impact extended far beyond the courtroom. It became a powerful tool for exonerating the wrongly convicted, bringing closure to families torn apart by uncertainty and highlighting the fallibility of eyewitness testimony and other traditional forms of evidence. Moreover, DNA fingerprinting found applications in identifying victims of mass disasters, reuniting families separated by war or natural disasters, and even tracing ancestry back through generations. Section 8. The Ethical Frontier, Navigating the Complexities of DNA. As DNA technology advanced, it raised ethical, legal, and social implications. Questions about privacy, discrimination, and access emerged. Imagine a world where your genetic information could be used to deny you employment, insurance, or even a loan. Or a future where parents could genetically engineer their children, selecting for traits like intelligence or physical appearance. The ethical dilemmas are complex and multifaceted. Who owns your genetic information? Should it be shared with employers or insurers? How can we prevent genetic discrimination? These questions require careful consideration and open dialogue. The future of DNA science is filled with promise, but it's up to us to navigate the ethical landscape wisely.
From the humble beginnings of my pea plant experiments to the groundbreaking discovery of the DNA double helix, the journey to unravel the secrets of heredity is a testament to human curiosity, ingenuity, and the relentless pursuit of knowledge.
It's a story that spans continents, disciplines, and generations. A story that continues to unfold today with each new scientific breakthrough.
Imagine the world before we understood the nature of inheritance. Traits were passed down from parents to offspring, but the how remained a mystery.
Diseases ravaged families, leaving behind a trail of heartbreak and unanswered questions. The idea that we could understand, predict, and even manipulate the very essence of life seemed like a distant dream.
But then came Mendel, a monk with a passion for science and a keen eye for observation. My experiments with pea plants, meticulously documented and analyzed, laid the foundation for the field of genetics, even though my work went largely unnoticed during my lifetime.
Decades later, the discovery that DNA was the genetic material, followed by the revelation of its elegant double helix structure, ignited a scientific revolution. Suddenly, the secrets of life seemed within reach. Section 2, DNA Today, a revolution in progress. Today, DNA technology is an integral part of our lives, transforming fields as diverse as medicine, agriculture, forensics, and conservation biology. We can now read the entire sequence of the human genome, identify genes associated with specific diseases, and even edit the genetic code with unprecedented precision. Imagine a world where diseases are diagnosed before symptoms even appear, where genetic testing guides personalized treatments, and where gene therapy offers hope for previously untreatable conditions. This is the promise of DNA technology in medicine, a promise that is rapidly becoming a reality. In agriculture, genetically modified organisms, or GMOs, are helping to feed a growing global population, while DNA fingerprinting has revolutionized forensic science, providing a powerful tool for identifying suspects, exonerating the wrongly convicted, and even reuniting families separated by war or disaster. The impact of DNA technology extends far beyond the laboratory and the courtroom. It's being used to trace ancestry, track the spread of diseases, protect endangered species, and even engineer bacteria to clean up environmental pollutants. Section 3. The future of DNA, unraveling the mysteries that remain. Despite remarkable progress, many mysteries remain. The human genome is far more complex than we imagined. We're only beginning to unravel gene regulation, the role of non-coding DNA, and the interplay between genes and the environment. Imagine harnessing DNA to eradicate diseases and engineer crops. These possibilities are now within scientific inquiry. The future of DNA science is filled with promise and challenges. We're grappling with the ethical implications of gene editing. The journey to unravel DNA secrets is far from over. Section 4. The ethical imperative guiding our genetic destiny. The power of DNA technology comes with a profound responsibility. We must proceed with caution, guided by a strong ethical framework. Imagine a world where genetic information is used to discriminate. The gap between the genetically privileged and disadvantaged widens. We must engage in open dialogue about the ethical implications. Establish clear guidelines for genetic testing and editing. Ensure equitable access to these technologies. Proceed with wisdom, compassion, and respect for life. DNA and the Symphony of Life. DNA connects us to all living things on Earth and across billions of years of evolutionary history. Imagine the vastness of the cosmos, filled with billions of galaxies. Here, on our pale blue dot, life has emerged in all its diversity, united by DNA. From microorganisms to whales, redwoods to orchids, every living thing carries the instructions for life in DNA. This fills us with awe and wonder. We are part of a grand symphony of life.